Well, hello there. Welcome to the Thursday seminar, and today I am excited to introduce our seminar speaker. So this is Dr. Nate Jue from CSU Monterey Bay. So Dr. Jue studied biology and creative writing at Colby College, and then he moved on to a PhD in biology from Florida State University, where he studied larval dispersal and population genetics of gag grouper. I had to look up that it was a fish because it was just called gag. Jokes. In the Gulf of Mexico, and then he transitioned from there into looking at larger scale genomic approaches in his postdoc at the University of Connecticut. And so since joining CSU Monterey Bay, his research now spans all sorts of different types of taxa and approaches. So there's a big span of animals, from fish to salps to humans and pygmy elephants, which is fun. And he focuses on using genomics and bioinformatics to study the connection between genotypic and phenotypic diversity, so sort of the connection there. And so his lab studies various projects, including bacterial, functional, and ecological genetics. They look at genomics and evolution of various marine species, including salps, like I said, the evolution of sex change in fishes, and the evolution of placenta and other interesting structures in fishes. But today, he's here to talk about some recent work on elasmobranchs and what he's learned from studying the genome of the great white shark. So please join me in welcoming Dr. Jue.
Thanks so much for inviting me. I'm really excited to talk about this interesting project that I got to be a part of. Before I start, I'm going to start off with a little bit of my infomercial part of it. So if you guys are interested in the kinds of things that I'm talking about today, I actually do teach a class as a graduate section for those who are graduate students here. But it's a class where we do a lot of the things that I'll be talking about today. We do learn about how to do those types of big data science, data analysis processes. And we're actually going to be using data from an international research project known as the Vertebrate Genome Project. Here's an example of an organism I'm working on right now, the spotted scat. I'm working on their genome. But basically, it's an effort to sequence about all the 70,000 vertebrate genomes that are for extant species. We're focusing about 260 right now, but it's part of broader collaborations with a bunch of different groups that are all working on building up reference levels, so very high-level genome sequences. So there's a lot of cool opportunities to learn some skills that would allow you to have some nice vertical opportunities into this area if you're interested. So if you are interested, just come talk to me about it afterwards. I'm happy to tell you about the class. Today I'm going to be talking about this study that was done, that we published last year in PNAS, or actually this year in PNAS. This was a collaboration between researchers at Cornell University, Nova Southeastern University in Florida, St. Petersburg University, Stephen J. O'Brien's group there, and then folks at the Monterey Bay Aquarium and me. So we have a bunch of them. A lot of this work was done by Nick here, Nick Maher, who's now a faculty member in Nebraska, and his advisor, Michael Stanhope. So a lot of thanks to them. And then we had, this is Sal Jorgensen, our local white shark biologist, who's also a large contributor to the project. Want to make sure right off the bat I recognize the people who helped support this project. So Guy Harvey Institute supported a lot of the initial sequencing of the white shark genome. And we got a lot of support from both Illumina and the Monterey Bay Aquarium, who donated a lot of materials and effort in helping us, and money in helping us actually get this work done. So big shout out to them. Without them, we wouldn't have gotten this project finished. So I'm going to start off with just a little bit of an intro into genomics, because I don't know how genomic-y you guys are, and you all might not be genomicists. So let's just chat a little bit about the context of the kind of data that we're going to talk about. In the end, I'm interested in doing what a lot of evolutionary biologists want to do, which is understand why we see the kind of diversity of life that we see, right? So in that process, I'm going to use genomic methodologies to examine those questions. So why is genomics a thing? Well, one of the reasons why genomics is a thing is that the cost of generating a genome sequence has dropped precipitously over the past 20 years or so, right? So the human genome cost about $3 billion to build. Now you can sequence a human genome for about $1,000. We've seen an exponential increase in the number of reference genome sequences that are in databases like NCBI. So there's been a rapid increase in the amount of references that we've been looking at and the number of species we can actually look at. So we're going to be taking a comparative genomics approach. If we just break down this word, we used to talk about genes as being the sort of the main thing that we were focused on when we talked about genetics, right? Here we're thinking about just a single unit of heredity or a distinct sequence of nucleotides. We then progressed forward into genomes where we're talking about the complete set of genetic material, right? We then think about genomics as being the branch of biology which is studying this complete set of genetic material, looking not only at the evolution but also the structure and the function of this complete set. And then if we put that in the context of comparative biology, which has historically looked for cross lineages, patterns of variation that can help us explain sort of the disparity and the diversity of life, we combine that into comparative genomics, and what we end up doing is using complete genome sequences to look at different species and trying to identify regions and components of genomes that are both conserved and vary, so both conserved and diverged among taxa, to try to understand what is the genetic basis for the differences in diversity of life that we see among species. Right? So that's kind of how the whole process breaks down sort of conceptually when we look about it. And the idea, I like using this quote from Goethe to sort of understand why we might be interested in doing this. So, he who doesn't know a foreign language does not know anything about his own. So this is the idea that we can't really know as much as we want to know, say, about ourselves until we start looking outwardly and understanding other solutions and other ways that organisms have adjusted and evolved. Right? So an important concept, I think, for us understanding why we do comparative work. For a specific example, classic example of where comparative genomics has been important for us in understanding a base component of biology is thinking about Drosophila melanogaster and the study of homeotic genes or Hox genes. Right? So in this case, we know that Hox genes, you might have learned this in like your intro bio class, right? Hox genes occur in a collinear fashion along a Drosophila chromosome, right? We have homologous genes, so the same set of genes that we find in humans. And by studying them in Drosophila, we learn that basically this collinearity matters. So if we take genes like Antipedia and we move it farther up in this group, suddenly they start growing legs out their eyes, right? So like it has a big effect on where development happens and how it happens. They're expressed in a way that starts at the anterior end of the fly and goes to the posterior. And we see a similar pattern, a conserved pattern happening in humans, right? And so we can see that by understanding this fly, we can learn a lot about the development of forms and what may be part of the development of form in and of ourselves. And we can also see how when things diversify away from this process, we can get additional function and additional components that may be contributing to that diversity. So just an exemplar of why this perspective is important. Additionally, when we think about what's in a genome and how genetic variation explains phenotypic patterns, we've got our central dogma, which is what you all learn in intro bio. We start off with DNA, gets transcribed into RNA, then it becomes a protein through translation, right? And we generally think that this is what drives the phenotypes that we observe in nature. This is what drives the organisms that we see, right? Proteins are the things that do functions. And what is responsible for this phenotype? Well, it's this genetic sequence which underlies this protein, right? This protein sequence. So we see this direct connection between the two. And that is definitely an important component of how organismal biology works. But when we think of what's actually in a genome, and we think about a genome like the human genome, only about 2% of our genome is actually coding sequence, is actually coding for a protein. So 98, 97% of our genome is not actually coding for a protein. About 80% of it is actually transcribed, so it's actually made into RNA, which may or may not have function. We do see that there are other things in the genome, things like pseudogenes, so copies of genes that may not be active anymore. They may have come from duplications of genes or from viral vectors. right? So there's other things going on, but a large component of actually what makes up our genome are these repetitive elements. So in the human genome, about, depending on how you calculate it, it's about 45 to 70% of the genome is made up of repetitive element. And what are these repetitive elements? Well, they're largely what they sound like. They're things that repeat a lot. And they can be different kinds of things. They can be genes that we have lots of copies of them, like globins and actins, or ribogenes. They can be things that don't have any sort of specific function, right? And so a lot of these things are often from viruses that have infected our genome and made lots of copies of themselves. And then we can have some highly repetitive stuff that are just like, repeated bases of AT, things like that. And sometimes they can be well distributed throughout chromosomes, and sometimes they're isolated. These kinds of repetitive structures are isolated to very specific parts of chromosomes that have very specific functions, like telomeres and centromeres. So these are the parts of a genome. Transposable elements are another important part of these repetitive elements, made famous through the work of Barbara McClintock. But these are often, we refer to them either as retrotransposons or DNA transposons. These generally come from retroviruses and from DNA viruses in their two cases. And we see them, this example in corn is one of these cases where these genes like to jump around your genome, like they can move around. And the reason why we see variation in the coloration of these kernels is that these genes have actually jumped to different places in the genome and either turned on or turned off this dissociator activator mechanism, which allows for whether or not anthocyanins are active or not active in kernels of corn. So you think genomes as being like this one stable thing, but it's actually a very dynamic process with lots of components that we don't necessarily always think about. It's not just proteins, it's all this other stuff that's going on and that can have effect on the makeup of an organism. So specifically, one of the reasons why I'm interested in comparative genomics is for using comparative genomics as a way of looking at adaptation. So what is the way that we can identify the types of reflections of evolution and how it's manifested itself in phenotypes that we can associate with a genetic origin? And so there's a couple different ways that we can go about doing this. We can do things like we can look at the presence and absence of genes, and so we can use as a metaphor for understanding this process. Think about like how a radio, like a stereo system might work. And so if we think about a stereo system, like if you want to have like a bunch of specific functions for your system, you can have amplifiers, you can have your head, which has got all your digital connections and everything else. You have different parts of the stereo that are similar to each other, may have been originated to different parts, but have now been sub-functionalized into different components. In this way, we can have the same thing for genes, where we can have multiple copies of genes, or maybe one gene interacting and being specialized with another gene. When we talk about molecular adaptation, we can talk about this as being changes in protein coding sequence. So we can have the presence or absence of genes, that being a way to get to an adaptation, a duplication of a gene, and then we can have changes in actual genes themselves. And so this can be like changing the frequency of a radio, right? So we're fine-tuning how proteins function and how they work. Gene regulation is another way that we could see adaptation occurring in organisms, and this is the analogy here is turning up the volume or turning down the volume, right? So in this case, we can control how much of a particular protein is made, and we can see in some cases whether or not a gene is turned on or a gene is turned off, and how that might allow for an organism to adapt to a different environment. When we think about genome projects, they're usually broken up into two types of data, so you can understand where this information is coming from. They're either genome sequencing data, which is the complete DNA content. When we remember, we're sequencing the entire genome. And this genome sequence shouldn't really vary from tissue to tissue. When we think about transcriptome sequencing, here, it's also known as RNA-seq. Here we're sequencing just the RNA from a sample. And in this case, like when we look at, say, kidneys, they're going to look a lot different. The genes that are active in kidneys are going to be a lot different than the genes that are active in epidermis and skin, right? So in this case, we're looking at the RNA that's identified these different places. When we think about how this relates to our ability to identify different types of adaptation from a molecular standpoint, typically genome sequencing is used for identifying things like gene presence, absence, and and you can also use it for identifying things like molecular adaptations to protein coding genes, because we just need to see what's in the genome, what is actually there. When we think about transcriptome sequencing, we can do this as well. We can look and see which genes are expressed and how they differ in their sequence, but we especially will use it for this type of gene regulation. I'm mostly going to focus on discussing results from the genome sequencing part of this project, but we have done a bunch of this transcriptome sequencing as well. So how do we identify something as being evidence for adaptation, right? So I'm going to talk about examples today where we're going to claim that we've identified molecular adaptation. Well, if we take sort of some expected outcomes in terms of what molecular data should look like, given if a population of organisms is either under stabilizing or directional selection, we can identify what that actually looks like at a sequence level and try to identify that case. So in this case, if we have stabilizing selection or purifying selection or negative selection, this means that there's some optimum that it's really not advantageous for an organism to move away from, right? There's some sort of perfect spot, and if they go too far away from it, they're heavily selected against. Directional selection, often referred to as Darwinian selection, because it exemplifies a lot of what Darwin talked about, and also referred to colloquially as positive selection, thinks about the idea that we move from one optimum to another optimum. So there's some sort of progression of genetic change that is happening at a rate faster than we would expect by chance, right? And so a good example here is we can look at alignments of coding sequence, and what we can see is we can find specific spots that are going to be very different among, say, one taxa like an elephant. So in this case, we find a few specific codon positions, protein coding sequence, coding for an amino acid, that we think are happening. We can see an accumulation of these, and we would predict that these changes have happened at a rate faster than we would expect by chance. So this is how we identify whether or not we think something is under positive selection, or in other words, we have evidence for molecular adaptation. So, it's a whole bunch of background. What about sharks? Things about supposed to be white sharks, right? So, these are the species that are out there, that were out there when we started working on sharks in terms of having genome sequences. There were only 4. That's not a lot. There's a few more that have come out since then, since we published the white shark genome. But we thought, we needed another shark out there, and why not do the white shark, right? Obviously, we have a white shark biologist who's very interested in white sharks, but they're a very charismatic species. But there's a whole bunch of other reasons why we might be interested in thinking about sequencing white sharks. They are, what do we know about white sharks? They're an endangered species. We tend to think of them, right, popularly, we're often thinking about them as the origin of shark attacks and Jaws and things like that. But they're incredibly interesting species in terms of their global distribution. We know there's multiple populations. They have a bunch of very interesting adaptations. But we didn't know very much about them genetically. We only had a little bit of evidence on what their patterns of geographic variation were in only certain parts of their genome. So there was a lot to learn by looking at these guys. Additionally, if we think about vertebrates in general, there's a whole bunch of bird sequences and mammal sequences and fish sequences. There aren't very many shark sequences. So if we're interested in learning about the evolution of vertebrates, getting some more representation in this part of the vertebrate phylogenetic tree is a big advantage for us. I'm specifically interested in sharks because I think they have really cool immune systems. If you think about it, right, these are cartilaginous fishes, so how do they make white blood cells? They have some very specific adaptations to do that, but they also have very different antibodies than what we have. So, our antibodies are these heavy chain, small chain antibodies. Sharks have these 2 heavy chain antibodies with the ability for some of these pieces to break off and form these nanobodies sometimes too. So there's some really interesting ways. They also have adaptive immunity just like we have. And so this is people are generally interested in antibodies for a whole suite of reasons. Evolutionary, this is interesting to me, but also from a biotech perspective, we use antibodies in everything from disease assays to pregnancy tests, right? So there's a lot of biotechnical things. Another reason people are interested in sharks is there's this idea that sharks don't get cancer, right? So there's people who've made some advancement in their career by talking about that, and then when people said that it wasn't true, they came back with a response, right? And then other people have other takes, right? And so everyone's trying to figure out, it's obviously an area of a lot of concern to people, but because of this kind of controversy, we might think that there's something that we might be able to learn from sharks. And so when we think about cancer, how does it generally function from a molecular standpoint, right? We have a cell cycle that a cell goes through when it goes through normal cell growth. We have these checkpoints that limit the process of cell growth, and cancer generally occurs when we have some breakdown in these checkpoints, okay? And if we think about it, cancer, we define it typically as a genetic disease where it's, cancer, it's cells that divide in an uncontrolled fashion. The reason why we call it genetic disease is that it arises from some types of mutations that we find that occur in cells, and it causes those checkpoints to fail, which leads to a change in the nucleotide sequence of the genome of these organisms in specific cells. And then those cells, if left unchecked, can go on to form tumors. And this, of course, this disease has affected many of us and the people that we love, and is a huge area of concern and interest and research in the U.S., and is an important area for us to think about. And so we started thinking about, well, why would we be interested? And one of the main mechanisms for this process historically has been things like tumor suppressors breaking down. So we have these genes that basically induce DNA repair mechanisms that come in that act as the gatekeepers on those checkpoints. And when they acquire mutations, then that whole process of keeping those checkpoints and not allowing a cell to reproduce when it's got a mutation break down and leads to tumor formation. And so a big process of what leads to cancer is whether or not these things are active or inactive when they should or shouldn't be. So why would we be interested in looking at something like a shark if we were thinking about cancer? Well, there's this idea out there. It's called the Pato paradox. And in the Pato paradox, basically, this was work done by Richard Pato, and who was a statistical epidemiologist who worked with zoos. And what they found was that if you looked at the size of an organism and its lifespan, so in other words, if we think of how many cells an organism might have, right, that might be the more cells you have, that would be more opportunity for cancer to possibly occur, right? Because you have more cells, there's more possibility that you could have a mutation in one of those cells that could lead to a tumor. And what did they find? They found no good relationship between the size multiplied by the lifespan of an organism and the occurrence of tumors, right? So they found there was no relationship. You notice the elephant out here had one of the lowest rates. And so it didn't seem there seemed to be no correlation there. So what is going on that these organisms are doing that's different than us? When we look at elephants in particular, they have a bunch of different solutions to this problem. But one of the big ones is that when we think about genes like those tumor suppressors I talked about, P53, instead of having just a couple copies, they have lots and lots of copies of them. So in other words, if they lose one, it's not going to be that bad for them if one becomes inactive because they got lots of backups. So that's one of the strategies that elephants have evolved to deal with this problem as a large-bodied, long-lived organism, right? They also have some specific molecular adaptations that they've evolved as well to deal with this problem. So let's get back to this guy. Let's get back to this shark, right? So, and back to this paper. So in our paper, we were specifically looking at how these adaptations could be associated, by looking at the genome, how these adaptations could be associated with things like wound healing and the maintenance of genomic stability. So can we look at their genome and identify components that we think might be contributing to the fact that maybe cancer isn't seen that often in these guys? And we think about a white shark, they are set up they have a lot of opportunity for there to be mutations, right? So they're large-bodied, maybe 20 plus feet at max, right? They're long-lived, can live a long time. They have large genomes. Their genome is about 1.5 to 2 times the size of our genome. So there's all kinds of chances for these things to have the kinds of mutations that we experience, right? But we tend not to see, it's not that we never see cancers, but we don't see them at the same rate as we see in other organisms. In addition to that, genome sequencing on these guys, we were interested in doing it. You know, they're regionally warm-blooded. They've got rapid wound healing. They've got all kinds of unique adaptations in terms of their sense of smell. We're interested in their conservation. And as I said before, I'm interested in their in the evolution of their immune system and where their phylogenetic position is. So a bunch of different reasons to look at their genome. Just so you know what a genome assembly is, I'm going to talk about a genome assembly, the results of a genome assembly. What is a genome assembly? It's an attempt to accurately represent an entire genome sequence from a large set of very short genome DNA sequences. So keep in mind, this is an attempt. This isn't the final version. In fact, I know there's another group that's actually doing a genome sequence of this organism right now, which will hopefully be even better than what we did. This is from a lot of data. This is from, how many billions of bases? I think this is almost from 1 trillion plus bases of sequence that we use to assemble the sequence. So it's a lot of data. And these sequences were all way shorter than what the actual genome sequences of this shark, right? So we have this difficult challenge of taking tons of data and distilling it down into this one genome sequence. And how this process basically works is we take a whole lot of sequencing data, we try to line it all up, we create these graphs, and then we can take these graphs and we compress them down to what we identify as a consensus sequence. And in an ideal world, this would be like what a chromosome is, right? This would be the sequence of a chromosome. In actuality, it doesn't. It's much more difficult to do that. And then after we've identified this consensus sequence, then we try to identify the parts that are on it. Where are the genes? Where are the repetitive elements? Where are all these other components? So if you can, a good metaphor, I think, is like thinking about doing a really hard jigsaw puzzle. So you got your regular jigsaw puzzle, but what if you didn't have a picture, right? And if you're lucky, maybe you've got a related species, so maybe the picture's fuzzy, but you still have no idea how many pieces are in it, so that makes it also hard. Once you start assembling it, you're like, okay, I kind of put it together, but then, I can figure out, okay, this piece should probably go over here and this piece up there because of these similar colors. So I can move them around and try to rearrange them. But when you have stuff that all looks the same, when you're just doing the water, it can be really hard to put it together, right? And that's the problem with a genome assembly, too. So parts that are repetitive are really hard to put together. Sometimes it's nice. You got like a nice feature that you can use to line things up, and you can do a really good job in that part. And then as you go through it and you do more and more sequencing, and you try to fill in as many parts as you can, and eventually you have to ask yourself, did I do enough? Is it good enough? And so we did a genome sequence, and somebody else is now doing another one. So obviously somebody else decided, hey, you know what, that's probably not good enough. And that's okay. That's part of the process. Even if you get all the way to this point, inevitably you're going to find that there's something that you dropped on the floor that's not in your thing. And sometimes you just try to jam a piece in where it's not supposed to go, just because it's got to go in that spot. So, it's really hard. And genome sequences never look like this. It's never this complete of a picture. It's very difficult to do. We almost never get to this point. And so just keep in mind that, this is the ideal. We're striving for it. So how did we do? We have a genome that we estimated to be about 82 chromosomes, about 6.3 gigabases long. After we did sequencing, we figured out that it's probably a little bit smaller than that, probably closer to 4.6 to 5 gigabases in length. And we generated a genome sequence which was about a little less than 4 gigabases, so we got about 85% of the genome in our genome sequence. So we feel pretty good about that. If we look at the contiguity of our sequences, we find that the N50 level, which is kind of like a median level of how big our sequences are, our scaffolds is about 2.7 megabases, so that's a million bases long. We estimated there are about 24,000 genes, which seems about right for a shark, and about 60% of the genome was identified as being repetitive. What we did then is we looked at other shark genomes, so whale shark and elephant shark, and we looked at our shark, white shark, our genome and our transcriptome, and we looked to see how many of these, there are these benchmark single-copy orthologous genes, so genes that we expect all metazoans to have or all vertebrates to have. And we said, well, how well did we do in recovering those, right? And it looks like we did pretty well for the most part. On average, we were, plus 90% for all these genes that we were looking for. There were only a few that were missing, which might be expected, but we were at least comparable to what had happened in other taxons. So it looks like we generated a pretty complete genome, at least from a gene perspective. If we think about the repetitive elements, what we can see, we had a lot of regions that were just identified as non-coding, so we weren't able to identify what that region was, but we just know that it did have coding sequence in it. We found there were a lot of line elements, so these line elements actually originally probably were remnants of viruses that infected this genome. And then we find that there were quite a few transposable elements, both LTR, so ones that came from retrotransposons, and ones that came from DNA viruses or DNA transposons, and then some fairly large unclassified group. So still lots of repetitive elements in there, and in terms of what their function are, we're not really sure what they are. But not a lot of evidence of segmental duplications or lots of simple repeats in this data. So this is probably kind of like if we take the similar perspective on like the metaphor of what it looks like, it probably looks something like this, right? In terms of like how well we did in terms of our assembly. So what about selection, right? We talked about this idea, can we identify adaptation? So remember we were talking about, can we identify genes that we think are evolving more rapidly than we would expect by chance? So this is that example of Darwinian selection, directional selection. So what we did is we found that there were, if we looked at the sharks, in general, the elasmobranchs, specifically, we can see that here we've got about 67 genes that we were able to identify that were under positive selection in sharks, 21 different genes that were under positive selection in whale sharks, 22 genes that we found to be under positive selection in elasmobranchs, and 17 that were associated with just elephant sharks. So these are genes that along this branch were evolving more rapidly to diverge away from homologs in other species than we would expect by chance. So what we did find is that it seemed that when we looked at sharks, a lot of the genes that we were finding that were under positive selection were involved in genome stability. So they're involved in DNA repair, DNA damage repair, DNA damage response, translesion synthesis, a whole bunch of things that were involved in repairing DNA or identifying DNA that had been damaged, right? And then we also found that there were a bunch of these gene ontological terms. So these are terms that basically explain what a gene does, right? So, and a lot of them were associated with DNA damage, repair networks. We found that a lot of these terms were enriched. So in other words, the things that we found in positive selection, under positive selection, we found these terms associated with those genes more than we would expect to by chance. So in other words, they had lots of these kinds of functional terms associated with them. And again, this is that example of that shift, right? We're seeing directional selection in these DNA genes, DNA repair genes, and what that means is that it seems that we have genes in this DNA repair network that are highly tuned, that are highly adapted to repairing DNA, right? So to that DNA repair response. And we can look at sort of broad scale. These are all the DNA repair or genome stability genes that we found that were under positive selection. And you can see they're doing a broad range of different functions. There are ones that are involved in scheduled cell death. There are ones that are involved in DNA repair responses. There are ones that are involved in DNA repair. There are others that are involved that are tumor suppressors and p53 regulators and things of that sort. So definitely these are, it seems in general, sharks, especially sharks with large genomes like whale sharks and white sharks, have a lot of adaptations for these adaptations for DNA repair in their genomes. One other thing that we noted was that here, if you see something in blue, that means that it's a term that's commonly observed. If you see something that's hot, it's a term that's rarely observed. So when we look at the terms that are commonly observed, the cooler terms are often associated with some of these DNA repair mechanisms again, right? So this DNA damage response, things of this sort, especially in sharks and whale sharks. Whereas we move over into something like an elephant shark, the genes that are under positive selection are not so much associated with DNA damage anymore. But that's an organism that has a much smaller genome. Their genome is less than a gigabase. Again, indicating that these sharks here with larger genomes have evolved specific adaptations to deal with DNA damage. This is looking at this MDM4, which is that P52 regulator that I showed before. And this is just showing you kind of like what a protein would look like and where these sites are that are actually under positive selection that we found on this protein. And what we find is that there's a bunch of sites that are under positive selection in these disordered parts of the protein. But even in this P53 binding pocket, right, there's a lot of fine-tuning of adaptation that has happened in that region. So again, this would indicate that they have evolved specific approaches to dealing with DNA repair for these larger organisms. So again, if we think about all of these repetitive regions, one thing we can understand is that a very small proportion of the genome of this organism is actually going to be coding sequence, right? The majority of it, the vast majority of it is going to be this repetitive element. And one of the things that we know is that repetitive elements tend to mutate at a much higher rate than non-repetitive elements. And one of the things that we found is in addition to these to these repeats, we found lots and lots of copies of various types of histones. Way more in our large genome species like whale shark and white shark than an elephant shark, which had a much smaller genome. And so what these histones do, right, is they bind, they wrap DNA around themselves, and a lot of times, especially histones like histone 2AX and H2AX and H2A, are involved in initiating DNA repair networks. So they can be used as ways to recruit DNA repair mechanisms that come in and fix mistakes. So we think that this expansion of these histones may also be one of the mechanisms by which sharks have evolved adaptation to having large genomes. When we think about some other cool things that are separate from this idea of how sharks have dealt with DNA damage or the opportunity for developing cancer, we also find some very interesting adaptations with regard to wound healing, where we can look at a gene like fibrogen gamma chain, which is involved in blood clotting, and we can find a whole series of very specific adaptations we only find in white sharks and whale sharks. We can also look at other things like how we can think about how sharks might smell blood in the water. We know that sharks have a very acute sense of smell. Interestingly, this is looking for copies of olfactory receptor genes, or OR genes. And what we can see is that for a lot of species, one of the ways they deal with having a better sense of smell is to increase the number of copies of these olfactory genes. Well, sharks haven't done that. Sharks, if we look at elephant shark, there's only one copy in their genome. And if we look at white sharks, we were only able to find basically 2 functional copies of olfactory genes. And so it looks like sharks are not their acute sense of smell is not coming from these olfactory receptors, but instead we think it may be originating from these vomeronasal vomeronasal systems. So here, it's a different way of sensing smell, basically. So sharks, instead of expanding their olfactory receptive systems, have expanded and generated more copies of genes involved in this vomeronasal process. So it indicates that this mechanism may be more important for shark smell than for sense of smell than the olfactory receptors. So that's a broad spectrum of all the different ways of adaptation. And you can think to yourself, well, okay, we learned all this different stuff. So we learned some cool stuff about, okay, sharks have like their own way of smelling that's different than what we have. I would bring us back to understanding like how we can actually use some of this information. So, this is just the sort of start of what this project is. Anytime you do a genome assembly project, it's a very inductive experience, right? So you're doing, it's discovery-based science. We're diving into an organism to learn something and discover something new, but there's still a lot of other things. This is just a starting point for us. And so, the next thing to do is to try to think about, well, how are these molecular functions and the gene content that we observe in these sharks, how is that going to inform us about DNA damage repair and wound healing in general? Can we then learn something about the specific adaptations, the specific genetic changes, say, in a shark, and use that to understand, say, maybe the risk of developing cancer in a human? Are we able to make that connection? Can we understand something about our mechanism by looking at their mechanism? And so that's some work that various members of our group are working on right now. We're also doing some work using this genome sequence to do a better job of looking at how different populations of white sharks across the world are different from each other. And that's part of a collaboration we're working on with Hopkins Marine Lab at the moment. And then lastly, I've been looking at some of the blood transcriptomes of these sharks to try to identify how their immune system is functioning and what are some of the novel ways that they may be addressing immune responses. And so there's a lot of fun stuff to work on in the future. We got to keep on making more Sharknado movies because they're awesome. And we're really excited. So just a little bit of a brief primer on some of the results from the study. And if you have some questions, I'd love to take them. So thanks.
So we know that the percent of non-coding genes is really low in most organisms, but given the size of the white shark genome, do we see that it's about the same percent, or is it even smaller compared to other organisms?
It's smaller because the genome is so big. So it's less than, it's 1% or less. So it is a much, they have all this, we actually aren't sure because it wasn't easy to identify all this repetitive element, because there are not a lot of other sharks we can use to identify it. So like, what are the viruses that infect sharks? Like, we don't have a great sense of that, right? So how do we find all the transposable elements? That's harder. And then they also had these, it seemed like they had lots of what we refer to as satellite DNA, where they had sort of like longer segments that are repeated a lot, but it was hard to figure out.
So the number of coding genes is about the same?
So humans have about like a little less than 20,000 coding genes. Sharks, our estimate was around 24,000, and we think that we probably did a pretty good job with that part of it. I mean, I feel pretty confident that we did a decent job of gene prediction, and that would match in line with what we've seen in other species, other lassobrings and such.
Yeah, I was just wondering, how many white sharks did you sample from to run the sequence? Did you do it on one organism, or did you have to do multiple sharks?
It's a good question. And when you do a genome assembly, it's always easier if you do it on just one organism, because you only have 2 strands of DNA, right? And so you're like, okay, I'm only going to have a couple places that are going to be different between these things, but then when I line them up, it's going to make my life easier. Unfortunately, the work that the Cornell group did was off of one organism. And then when we decided to add in a whole bunch of other stuff to improve their assembly, The DNA we were able to isolate from the sample they had left was not at the quality that we could use to do the next part. So it was actually from 2 individuals.
Is there significant, I was looking at the GO-term plots that you had up there, and the white shark plot looked like the GO-terms clustered more. Is there some relatedness to
So the
GO-terms on the axes, or what is that?
Yeah, the clustering should be about whether or not those terms have similar function. So when they cluster together, so notice like a lot of the cold things clustered together, and a lot of them were DNA damage repair stuff. So that's what that was further evidence to suggest that DNA repair is more commonly observed in the positively selected genes. They call that, what do they call it? They call it like something space, like verbal space, or semantic space. Thank you very much. Yes, that's exactly right. Chris.
So you talked about DNA repair, but what about wound healing?
What about it? So
You didn't really address that in the talk, but
yeah, so
remember from the paper.
What we found from wound healing is that, again, we found specific adaptations in a bunch of genes that are involved in angiogenesis and the recruitment of angiogenic growth factors. So it looked like there was some adaptation there. And then also, I didn't talk about this a whole lot, but if we look at the full complement of genes that we identified in white sharks, it looks that white sharks are enriched for specific categories of genes. So they have a lot of DNA repair genes in general as compared to another species. They also have a lot of angiogenic and angiogenic growth factor, wound repair type genes. So we found evidence for adaptation at a molecular level for that as well. Someone once referred to them as like the Wolverine of the sea. So, next one.
So, taking the comparative approach, can you look at other vertebrates of similar size and longevity and predict that they would also be enriched for DNA repair?
Yeah, so that's something I'd really like to do, right? So for large sharks, we're really interested to see whether or not the kinds of solutions we see, like the whale shark genome was, the one we were able to look at was not the best genome assembly. So it was an okay comparison for some things, but not, I think there's a newer assembly out that we could look at that would be a better comparison. But we're interested in definitely looking at large sharks, but also other large mammals, right? And so there is a blue whale genome that's going to be coming out soon through the Vertebrate Genome Project, and we're very interested to look at that. My guess is that the whales are probably going to do some things similar to the elephants, right? Because they're not that distantly related. So my guess is, if I had to make a guess, I guess that they would be increasing the number of tumor suppressors, which is something we don't see in sharks, right? We don't see an increase in tumor suppressors. Instead, we see these pathways of positive selection being the driving force of DNA repair mechanisms.
Cool. Thanks, everybody. And if you have more questions one-on-one, then we have a happy hour coming up after, and you're able to stay for a little while later. So yeah, feel free to get up, mingle, and ask